Okay, thank you for the invitation. And, our talk concern a much less defined project and program for caffeine treatment in GNAO1 related movement disorders. This morning we learned a lot of things about the background of the caffeine for the treatment of movement disorders in GNAO1 gene defect, but just to make simple, a complex matter, we can say that this complex postsynaptic network act on control, modulate the synthesis of CMP through the effect of ADCY5, and seem that the increase of synthesis of CMP may be related with movement disorders and may be also with the paroxysmal movement disorders in patients. As a matter of fact, many proteins involved in this pathway, when alt or gene, when altered, result in movement disorders, often paroxysmal movement disorder. But the problem is that movement disorders is difficult to be classified in patients with GNAO1 defect, as what discussed before with Dario about how to classify symptoms in a GNAO1 defect is have not a simple answer, because patients may present hyperkinetic movement disorders, hypokinetic, and very often patients suffer from hyperkinetic movement disorders and hypokinetic movement disorders. And also there is something which go over the problem of excessive hyperkinetic movement, something concerning with the learning of movement, most of all in very young patients. So at the end, we obviously should focus on hyperkinetic crisis. It was said before Dario, it's right to concentrate on this condition to be really a life-threatening condition for patients with GNAO1 defect. And this slide summarize what we already know, that caffeine is able to inhibit adenosine receptor, and adenosine receptor inhibition may result in a reduction of synthesis of CMP. In this way, we have a way to test the effect of caffeine, and also we have the possibility to control hyperkinetic movement disorders. But caffeine has other effect on GABA transport and release, but also high dosage may also act on the phosphodiesterase, but at dosage much more higher to what will be proposed for clinical setting. Caffeine is a well-known substance. Its advantage to be quickly absorbed by intestine, and also caffeine gate brain rapidly, and the concentration of caffeine in brain parallels the concentration of caffeine in blood. Also, half-life of caffeine is quite short, also although variable, a bit variable. And another interesting point is that the dosage of caffeine resulting in caffeinism or lethality is very far from the dosage we will adopt in clinical settings. So this is some advantages of caffeine as possible drug. There is many clinical trial involving caffeine have been conducted or are ongoing in pediatrics and pediatric medicine, and many other in adult medicine. As formulation, caffeine is available as caffeine base, as caffeine citrate, and as caffeine combined with different nutrients, amino acid, other possible metabolic substances, and dosage is extremely variable, starting from 1.5 milligram kilo body weight and to 10-15 milligram body weight, and tolerance of caffeine for by neonate is higher until 20 milligram kilo body weight. Okay, the interest for caffeine was generated for the paper on ADCY5, French paper on ADCY5 defect, and this paper was a retrospective study, but showed an impressive response of movement disorders associated with ADCY5 to caffeine. And another interesting point that adverse effects were really mild and mainly associated with higher dosages of caffeine. So this is a really preliminary interesting study. It was not a controlled study, as I said before. It's a retrospective study, but it's interesting interesting for our decision to adopt caffeine for treatment of GNAO1 patients, and also cellular exploration of cellular patients of variation of ADCY5, which are the gain of function variation in cellular, showed that effectively caffeine is able to reduce the synthesis of CMP. So there was a preliminary study about the effect of caffeine on movement of altered movement in a preclinical model of GNAO1 defect, and I would say that the first of this kind also activated a lot of physicians and also parents to adopt caffeine, sometimes to look for a possible effect of movement disorder with patients or child. And caffeine, also the study confirmed that caffeine acts through the effect of A2A receptors, and but a more recent study confirmed this therapeutic effect, but maybe also show possible alternative effect of caffeine in reducing aberrant motor alteration in the preclinical model. And the other study also we have the opportunities this morning to learn confirm the effect of caffeine for movement disorders in GNAO1 preclinical model. So speaking about patients, the problem of patients are mainly hyperkinetic movement disorders and hyperkinetic crisis, because this kind of disorders, when present, may be life-threatening for the patients, or in any case may result in hospitalization, sometimes in intensive care. And obviously, our target is to look at possible home intervention, pharmacological intervention to reduce hospitalization, to reduce the progression of hyperkinetic movement toward a hyperkinetic crisis. This is this girl shows what happened with an hyperkinetic crisis, and this is a severe hyperkinetic crisis that resulted in this condition in really protracted hospitalization and finally, unfortunately, in a neurochirurgical intervention with pallidotomy. Concerning hyperkinetic crisis, we decided to design a phase 2 open clinical trial of caffeine citrate in patients with defect, with general defect and dyskinetic crisis. Quindi the target is to look for home medicaments, medicaments that parents may adopt at home with very low risk, or also with the possibility that it may don't work. So we have a number of points to be clarified. Primary endpoints have to test the effectiveness of caffeine as a home tool, but also we have to explore what is the target of the effect of treatment concerning duration of the hyperkinetic movement, the frequency, the severity, and all these are variable to be explored during the trial. And there is a lot of secondary endpoint. Those findings is really important. We are convinced that there is an inter-individual responsivity to the caffeine. We should explore personalized treatment through a dose-finding approach. And another point is it's possible that caffeine may prevent this hyperkinetic crisis, or we should use caffeine for the hyperkinetic crisis when it is going to develop. It is another problem we have to face. And safety, caffeine, we know well the safe profile of caffeine, but we are not sure that patients with GNAO1 defect may not have a vulnerability to this substance, to this medicament. So you have to test safety also in clinical context. And also another important point is the persistence of the response to caffeine. After the first or the second of continuous administration, we should verify that the effect, the therapeutic effect is maintained in time. So the design is a dose escalation design, starting from a very low dosage, such as 2.5 milligram of kilo body weight day, until 10, in some patients, 20 milligram per kilo. And for the trial, we preferred the caffeine citrate instead of caffeine base. Among the other positive characteristics, the caffeine citrate has an excellent safety profile, as it is approved for neonate affected with apnea of prematurity. It offers a quicker time to peak concentration with respect to caffeine base, and also a high level of concentration in blood and in brain, and also half time and a clearance from blood quicker than caffeine based by 3 to 6 hours. This is The trial consists in three stages. Pre-treatment evaluation focusing on characterizing the frequency and severity of this kinetic crisis in each participant. This is the most important part of the trial because there is extreme variability in the frequency, duration, severity of hyperkinetic crisis in different patients. We have to establish a rule for each patient enrolled for the study, and parents, caregivers, will use, they are able to use the questionnaire and also to have instruction for video recording of the patient, because the effect of this treatment will be tested at home, and we will work for verify the effectiveness of this treatment at home, not in hospital, not in a condition where we can directly control the effect of the treatment. And this part is important. Also, the training of the parents, the training of the caregiver to share the same evaluation of a movement disorder they observe is really important. It should be precede the onset of the trial. And this is a sort of flow chart for the acute treatment. It's quite simple. Theoretically, we start with 2.5 milligram kilo for body weight, and when the crisis with the hyperkinetic manifest. So with the onset of hyperkinetic movement or increase of basal hyperkinetic movement, this dosage of caffeine will be administered, and we should have a period of observation by 40 minutes, and we have two possibilities that the drug, this dosage works. In this case, the drug will be adopted for the following episode, or it doesn't work. And in this case, during the following episode, the dosage should be increased to 5 milligram per kilo day, and so on for each repeated episode. And obviously, if the drug doesn't work at the moment of the specific situation, all the canonical treatment that should be applied, drugs, hospitalization, what you need, usually is adopted for the patients, for the child when he suffers from hyperkinetic crisis. At the end of this trial, we should decide if caffeine is working in these patients and is not working, and what dosage, what specific dosage for and specific patients. And the following stage include all subjects from the previous study, whether they were responsive or unresponsive to the acute administration of caffeine. Also in this case, by dose escalation approach. In this case, the aims is to test if caffeine treatment may prevent the occurrence of limiting the severity of hyperkinetic crisis. And so also in this condition, the treatment is preventive to the patients. The child started to be treated with 2.5 milligram of caffeine per kilo body weight, and it is for a period of observation. How long should be this period of observation? We have estimated that this period should not be longer than 3 months and should tendenzially be shorter, but in any case we should not go over 3 months, but maybe that our period of observation we have evaluated by the pre-treatment observation must maybe of 1 month, 4 weeks, 2 months. Any case, the pre-observation should be personalized. So at the end of this period of observation, if a relevant reduction is being observed, the frequency, duration, and severity of hyperkinetic crisis, the trial identified specific dosage for the patients. Otherwise, if it fails, in this case, the next dosage will be adopted, 5, 10, 15, and for the chronic administration of this part of the trial, we should not go over 10 milligram of per kilo of body weight. So this is the second part of the trial. What timeline of this trial is the ethics committee is spending. Patients' enrollment will begin in September. I would say that this project is not funded. The Chiesi pharmaceutical company has donated caffeine citrate for the pilot trial. Obviously, we applied a different grant in the hope to be able to extend the experimentation, and also we hope that if the caffeine showed be effective in improving condition of our patients with GNAO1, we can call for to EMA for approbation of this drug as orphan drug. Thank you. Thank you for your attention.